Right, so over to Jordan Cuff from Newcastle University to talk to us about networking nutrients and how nutrients could be a driver of invertebrate interaction. So over to you, Jordan.
Brilliant. Thanks so much for having me. It's a pleasure and an honour to be here, and thanks for all of you who've joined us now and hopefully watching a recording later for taking the time out of your day to sort of engage in all of this work that we've been doing. So just a brief summary about me. So Kieran's mentioned a few things about me already, but I'm currently a lead of the Foraging Ecology Research Group. So that's a research group that I've been building in Newcastle University. I'm a NEWACT Fellow, so that's a Newcastle University Academic Track Fellow at Newcastle University. I'm a Fellow of the Royal Entomological Society. For any of you who aren't members or fellows, I'd 100% recommend signing up. It's one of the best things I've ever been involved in. And I'm also Editor-in-Chief of the Royal Entomological Society journal, Agricultural and Forest Entomology, which I'm more than happy to talk about with anyone who's interested in submitting any papers or anything like that. Most of my research linked to the Foraging Ecology Research Group focuses on foraging of invertebrates particularly, so that's the trophic interactions that invertebrates engage in and the things that drive and constrain those interactions, but also the consequences and feedbacks and buffers of those interactions. So I'll be talking mostly today about a specific driver of trophic interactions, which is nutrients, but you'll pick up on a few other bits that come up that sort of cross into those other sort of determinants and consequences as well. So I'm going to first talk a little bit about invertebrates and ecological networks, just to sort of give a bit of background and summarise what that means and how we look at that. I'll then talk a little bit about biocontrol by spiders and the nutritional dynamics that they're constantly engaged in out in the field, particularly in this case in arable fields. I'll then talk a bit about integrating nutrients into ecological network analyses, so how we can bring nutrients into that whole food web context. And then I'll show you a few things where we've put these nutritional networks to work. So just to kick things off, I've got this nice cartoon that shows all these different invertebrates doing different things. So hopefully I'm preaching to the choir here, but invertebrates are incredibly important and all around us and constantly engaged in all sorts of different interactions. So here we can see examples of herbivory, where these caterpillars and hoppers and aphids are feeding on these leaves. We've got parasitism, where we have a parasitoid parasitizing a caterpillar. We've got predation, where we have ladybirds, spiders, and other spiders all eating other invertebrates, and we have pollination as well, with bees and butterflies visiting these flowers. And all of these interactions, importantly, form a complex web of interactions that cascade through whole ecosystems. So here you can see what we call an ecological network, or I suppose technically two. So you can see we have these circles, these nodes, which are all of the different species in this particular ecosystem, and these lines between them that essentially tell us that those are interacting. But it's often very complicated. We have crossover between different types of interactions, and we have all of these different organisms forming a really complex network that can go sort of miles and miles and span not just one ecosystem, but essentially the entire globe once you really start to connect everything. But a lot of the time in entomology and ecology, we focus on specific interactions. So in this case, we have a spider that's interacting with this hopper that might be a crop pest, for example. And we often focus on these because they're of some kind of economic significance or otherwise something that we deem to be important. So when we do focus in on these, we can start to study how invertebrates are contributing to our ecosystems and the ecosystem services that we depend on. So I wanted to highlight this wonderful paper from Radek Macalko et al. In Global Ecology and Biogeography, where they essentially showed that just having spiders there, and particularly diverse assemblages of spiders, can really have an effect on the density of pests in crops, but also things like the yield of the crops and damage to those crops by pests. So just the mere presence of these invertebrates is super important, but understanding the drivers of those interactions is really key. And importantly, as you saw in that previous network, these interactions aren't isolated. They form part of a wider network. So in this case, the spider's not just eating the aphid, it's also eating a few other things. So in this case, a cricket and a parasitoid wasp. So it might not always be providing an ecosystem service, it's doing other things as well that might sometimes even be a disservice. And importantly, it's also competing with other predators. So earwigs and wasps and beetles are going to be competing with these spiders for their prey, which can change the frequency of those interactions and sometimes whether the interactions happen at all. And all of these predators are, of course, eating other things as well. So this cascades throughout the entire ecological network, and they're also eating one another. And we even have things like parasitoids that might come and parasitize our predators. So it's a really, really complex picture of different things that might be driving these interactions. And for us to really understand why these interactions with crop pests, for example, are happening and how much they're happening and the things that drive them, we really need to begin to understand all of these wider interactions as well and how they impact that. But if we think about when we're choosing what we eat and whether to eat specific things, there are a series of questions we might find ourselves asking. So one of the first ones will be what's available. So if we have this bundle of food, perhaps the supermarket sold out of watermelon and avocado and apple, and suddenly the number of food items that we have to pick from is reduced. Let's say we're in the mood for something warm because it's a bit cold outside. Up here in Newcastle, it's definitely a little chillier today than it has been recently, so maybe you wouldn't want any of those sort of cold bananas or lettuces. So obviously an important part of what we eat as well is whether it's nutritious. So perhaps burgers and pizzas aren't always the best options, and suddenly we have quite a restricted range of things that we might eat. And all of these rules, importantly, in some capacity apply to insects as well. They're choosy about what they eat because they're similarly having to balance all of these different demands. And sort of drawing on that last example of nutrition, there's some really compelling evidence that nutrients drive these predator-prey interactions in invertebrates. So these are a couple of figures from a paper from Mainz et al. In Science in 2005, where they essentially showed that when invertebrate predators are fed a lipid-rich diet and then given an option between protein-rich and lipid-rich food, they'll always go for, or largely go for the protein-rich food, and vice versa. So we can see that they're sort of balancing their diets a bit like some kind of strict dietary regime. They're looking after themselves. But importantly, most of the evidence that we have for theories like this is based on lab feeding trials. So a lot of the time that'll be pictures like the one on the left, where we have sort of insects trapped in containers together in a sterile conditions, which can allow us to really get down to the mechanisms and some of the real sort of gritty details of these interactions. But it's not always necessarily a perfect alignment of what happens in nature, where of course things aren't happening in plastic containers and we can't necessarily control for all of the different competitions and sort of predators and risks involved in those interactions. So if we are to begin to understand what's going on in nature, instead of depending on these lab studies to know what these invertebrates are doing, we need a few bits of information first. So we can think of this almost like a menu. If we know what's available to the spiders, so the things that are on the menu, the nutrient contents of those things available to them, a bit like sometimes if you're scrolling through a menu these days, they'll have the number of calories in each meal. But we also need to know what that spider has chosen from the menu. And with those three pieces of information, we can start to work out whether nutrients are dictating what these spiders are eating. And I'll talk through that in a little bit more of a granular way in the next few slides. So a lot of the data I'll be talking about we collected a few years ago in South Wales. So the picture in the middle there is a nice field of barley just near Cardiff Airport. For those who've been to anywhere near Cardiff Airport, you'll know that that's quite exceptional weather. A lot of the time it's a lot drizzlier, but we were going around collecting money spiders mostly, so Linyphiidae, the really tiny sheep weavers you see that often balloon onto you in the spring and autumn. And alongside these spiders, we're also collecting vacuum samples of all of the prey available to the spiders in the places that we found them. So this is essentially just a leaf blower stuck on reverse setting. So you can see a first-person perspective of using one of those on the right. So we're basically hoovering up all of the community of prey available to these spiders. And once we got the spiders back to the lab, we used a technique called DNA metabarcoding to essentially take the DNA from the spider's guts and sequence it to identify what these spiders are eating. And I won't go into detail about that today. I'd be more than happy to talk about it another time, but I'll stick to the nutrients for now. But just based on that dietary DNA data, we can immediately start to pinpoint some of this biocontrol activity going on in these spider communities. So here you can see on the bottom the 5 genera of spiders that we looked at, so Bathyphantes, Erigone, Microlonifia, Pardosa, and Tenuophantes. And we can see the sort of purple blobs are the predators that they were eating, and the green blobs are the pests. So hopefully you'll see that most of the time the green blobs are slightly taller, so generally the spiders are eating more pests than they are predators, so they're doing good stuff for our ecosystems and our agriculture, importantly. But you can see that varies massively between different spider groups. So Bathyphantes and Tenuophantes are particularly good at eating pests, whereas, for example, Pardosa is maybe doing far less. And we can compare that between life stages as well. So what we found in this particular study is that the juvenile spiders are pulling their weight. They're eating loads of pests and very few predators, whereas the older spiders are maybe slightly less helpful, which is really good to know. So we can then sort of use that to help manage these spider populations in the field by perhaps trying to encourage habitats for spiders to bring new spiderlings into the world. But it doesn't really tell us much about the sort of mechanisms driving those interactions. We know the interactions are happening, but are they just eating whatever's available? Are they just going around and scooping up whatever they find? And I thought I'd use a little analogy focused on going shopping to sort of illustrate how we might be able to look at these choices the spiders are making. So if you went into Tesco to buy a sandwich, and they had 8 different sort of packets of sandwich on the shelf, you might ask yourself, which sandwich should I choose? And maybe one of the obvious choices is just to go for the most common one, the one that's most available. It'll probably be the first one you find anyway, just by probability. So in this case, it's the some leaves in bread sandwich. You can tell I took some creative liberties with the types of sandwich that Tesco provide. But maybe if you're just picking one at random, it won't necessarily be the most common. It could be the second most common sandwich, which in this case is the vegetable oil and disappointment sandwich. But you know, maybe that doesn't sound very appealing. Both of those flavors sound a bit drab. So perhaps you have a penchant for ketchup, and let's pretend for a second that Tesco do a ketchup sandwich. Suddenly you've got an obvious choice from the shelves, and a bit like that, we can actually start to assess, based on the availability of different prey, what spiders are choosing to eat, and perhaps the mechanisms driving those choices. So I mentioned already that we've done this DNA sequencing to find out what the spiders are interacting with. So we end up with a nice interaction network like the one you can see in the bottom left there. So we know some of the interactions these spiders are engaging with. But if we have the prey community data, that, much like the shelf in Tesco, tells us what's actually available to the spiders. And we can use some fancy computational modelling called null modelling to essentially tell us what the spiders are likely to eat if they just ate the things that are most available, so the most abundant resources or prey. And based on this, we can then compare the observed interactions that we get from the DNA with those simulations to find out what they're eating more or less than we would expect, just based on random foraging. And just to kind of run you through an example of how that works, if we have a spider with loads of different thrips individuals, a few aphids and a single fly, if we were to null model those interactions, we'd expect the spider to probably quite a few thrips, not many aphids, and probably no flies. So if we then did the DNA dietary analysis and found that they're eating thrips and flies, we can probably say that they're eating thrips about as much as we'd expect. We knew that they were likely to be interacting with them, but they're eating aphids less than we were expecting. So we can call that avoidance. So they're for whatever reason avoiding eating those aphids. And then if they're eating the fly, despite there not being many of them, it may well be a preference. So they're seeking these flies out in the environment, or otherwise somehow predisposed to eating them. So if we do that across a whole data set of loads of different interactions, we can build a really complex picture of the preferences of these spiders. So the main thing I want to drive home with this slide is there's a lot going on, and hopefully that's the impression you get. But we can see on the left some of the different preferences of the spiders. The blue dots are the blue dots are where they're avoiding different prey, and the red dots are where they're sort of preferentially seeking them out. And you can see in the network we have the blue links that show those avoidances and the red that show the preferences. So what we can basically take away from this is that the spiders are actually very picky. They all have different preferences, and they vary between life stages, sexes, and different taxa as well. But how do the nutrients in the prey actually affect these choices? Are they making decisions based on sort of balancing their nutrient intake? And to answer this, we needed to do a bit of protocol development. So I developed a protocol for analysing the nutrient contents of invertebrates. So this basically involves using that vacuum sampling to collect the invertebrates, crushing them up into tiny pieces, and then using some biochemical assays to work out how much lipid, carbohydrate, and protein there is in each invertebrate, using sort of what we call colorimetric assays. That's where different reactions produce different colours. Most people may remember them from school if you've not done them since even. So from that we can start to get this profile of what each individual invertebrate contains. So here we have a mock-up of what a single aphid serving might look like to a spider. These values are grossly sort of misrepresented, but just to give you an idea of sort of an anthropocentric perspective on what those data might look like, we can start to balance those fats, proteins, and sugars. So with that, we have the spider dietary data from the DNA metabarcoding. We have the prey availability from that suction sampling, and now we have the prey nutrients as well. So we know all three of those things that we decided at the start that we needed to know in order to identify whether they're foraging for specific nutrients. And bringing some of those bits of data together, we can start to look at the nutritional niches of different spiders. So here's a ternary plot. Most people absolutely hate them, but I'll just briefly explain how to read them, because there'll be a couple of these. Points that are closer to the top are basically richer in carbohydrate. Points that are closer to the bottom right are richer in protein, and points closer to the bottom left are richer in lipid. But the important thing here really is just that the different coloured points are in slightly different places. Particularly the yellow points, which are Pardosa, the only wolf spider genus that we looked at, are all clustered toward the bottom. So we can see that wolf spiders quite like protein and lipid-rich diets and not much carbohydrate. So already we can start to see, just by comparing the prey that they've eaten to the nutrient contents of those prey, what kind of things these spiders are likely to be looking for. We can compare that based on sexes and life stages as well, and you can probably see from the shapes that there's a bit more overlap, although generally speaking the male spiders tended to go for more carbohydrate rich prey, and the adult spiders did as well, although it can be a little hard to see with all those points in this case. But obviously the question we asked going into this is whether they're actually choosing things based on their nutrient content. Again, we've just been looking at what kind of nutrients they're taking in and what they're interacting with. So if we use the prey availability data to again predict what we think that they're likely to interact with, and look at the nutrients of those versus what they actually are interacting with, we get something like this plot on the bottom left. So the green points together in the middle are what we expect the spiders to eat in terms of nutrient profiles, and the purple points that are clustered much further around those green points are what we actually see them eating. So this shows that the spiders are eating a much less nutritionally balanced diet than we might expect. And this is actually probably what we'd expect to see if they are so balancing their diet, because with this DNA analysis we only get a small snapshot of what they're eating. So this could be these ones that are eating carbohydrate-rich prey, for example, might have previously been eating quite a lot of protein-rich prey or lipid-rich prey. So we may be seeing that sort of dynamic balancing of nutrient intake across this population of spiders. And again, we can look at those preferences across different groups. So in this case, each of the different points is a different prey. So you should see they're in approximately the same places in every plot. But if it's red, it's a prey that these spiders are preferentially seeking out. And if it's blue, it's one that they're avoiding. And the key thing to take away here is we have Pardosa, again, the only wolf spider, doesn't have many sort of distinct preferences. It's just kind of eating most things as much as it finds them. Whereas the web-building spiders, the linyphiids, all have different preferences. So Origone and Microlinifia have these big red blobs at the top, so they have preferences for quite carbohydrate-rich prey, whereas a lot of the others tend to be going for more kind of protein-rich prey. And again, if we compare between sexes and life stages, we see some really cool things, particularly that male and adult spiders tend to go for more carbohydrate-rich prey, so preferentially, of course, whereas the female and juvenile spiders are going for more protein-rich prey. So clearly there's some interesting stuff going on in these spider populations, whether that's because the juvenile spiders need that protein to grow, whereas the adults need the carbohydrate and the energy to go and find a mate and to forage. So some really cool stuff. But there are still a few gaps in what we know from all of this. So we don't really know how other interactions might be affecting the sort of nutritional choices that these spiders are making. We don't really know how these nutrients are flowing through the wider food webs either. And we don't really know how these nutrients might be driving that biocontrol interaction that we talked about right near the start. So that's where integrating all of these data into nutritional networks comes in. So we can start to build a more complex picture by bringing all of this together into a network. And just to briefly explain why that might be useful, so a nutritional network gives us a community level perspective. So rather than just looking at individual interactions, we start to see how that cascades across whole ecosystems. And that tells us how these nutrients are structuring food webs, so driving those interactions, but also the sort of indirect interactions that come out from that. And we can then start to look at how nutrients might be driving ecosystem services like biocontrol. And there are a few examples of people using network approaches to look at nutrition. So my favorite one is Timberlake et al. 2022 from People and Nature, so you can see that on the left. And this is where they looked at plant pollinator interactions in Nepal and how those might be driving the availability of different nutrients to the local population. So you can see at the bottom we have the pollinators, in the middle we have the crops that they're pollinating, and at the top we have the different nutrients that come from those crops. So it's a really nice example of how these nutritional networks can really tell us about the importance of these different services that the insects are providing. And there's another example on the right there, sort of looking at different food availabilities. But there are a few different ways that you could build a nutritional network, and we played with a few different ideas, particularly early on. So we looked at sort of coagulating or aggregating species together based on similar nutrition. So we can, instead of looking at species within this network, we can look at sort of nutritionally similar taxa together. And we called this sort of tropho species, which didn't really take off. It was quite complicated and quite difficult to see the trends. We could also look at nutrients as nodes. So on this right-hand side network, we have three different sort of coloured circles, which each represent a different nutrient, and each of the green squares is a different spider. So we can see, rather than their interactions with different prey, just the nutrients alone. But actually it's really hard to get any ecological information from that. It sort of oversimplifies everything. So instead we looked at integrating the nutritional data into the links themselves, so these interactions in the network. So just to remind you, we have the circles, which are the nodes. At the top of this network, we have the consumers, so that would be the spiders in this case. Then we have these lines that link them to the bottom circles, which are the prey, which are the interactions. And typically those links, those lines, the thickness of them depends on the frequency of interaction. So if the line's thicker, it means that there's more interaction between those two organisms. But we can instead look at the flow of nutrients between those organisms as those sort of line widths, those weights. So we can take the frequency of interactions and multiply it by the different resource nutrient contents to get different lines and different thicknesses depending on the different nutrients that are being transferred. And when we do that, we get something like this. So this is the idea of a nutritional network. So some of this is just sort of cosmetic information. So you can see we have these pie charts at the top, which are showing the mean nutrient content of the things that the spiders are eating. And at the bottom, we have the nutrient content of their prey in those pie charts. And then these lines, which are the things that we're actually analyzing when we do network analysis, are essentially based on the flow of nutrients between the prey and the spider. And if you want to read more about this, there's a QR code there, and I think there'll be a link in the chat to our paper in Journal of Animal Ecology that talks about this concept in great detail. But we can start to look at some of these really interesting ecological patterns using this approach. So I mentioned spiders and other invertebrate predators choosing what they eat based on nutrient content. So we can start to look at that process that we call nutrient-specific foraging by looking at the distribution of nutrients across the network. And we can look at all sorts of other different sort of hypotheses and patterns, like whether there's limits to some nutrients in the network that might drive completely different network structures, and whether the species eating things as a generalist or a specialist will completely change the network as well. And I'll talk through some of the examples that we can look at in a bit more detail now, by first showing you what a nutritional network actually looks like rather than these sort of cartoons. So all of the data that I've shown you so far with this spider prey network looks something like this. If we have the 5 different spider genera at the top and all of the different prey species at the bottom, and you can see it looks like something you'd look at with 3D glasses, but I promise you it doesn't look any more sensible with that, although I would like to experiment with looking at it like that. But essentially you can see there's lots of red going around, so that means there's lots of protein flowing through the network, lots of blue, so lots of lipid, and a little bit of yellow, which is the carbohydrate. But importantly, these networks often look quite chaotic, and it's quite hard to tell just from looking at the network what's going on. The real magic actually comes from doing the mathematical analysis on the links and all of the interactions. But what if these spiders are just randomly choosing things? They're not actually selecting nutrients intentionally. How can we test for that? So if we take some of the nutritional data that we've got, which just to give some sort of dummy examples here, we've got thrips, aphid, and hopper. One thing we can do to test whether nutrients are actually an important thing structuring these networks is by randomizing the nutrient contents across the different prey. So I've referred to this as shuffling the deck of nutrients. So we can literally just swap those nutritional values around randomly, and that will give us a completely different network. And if we find that the structural properties of the network are similar, then it might well be just random. So if we do this process of swapping the nutrients and rebuilding those networks, so thousands of times, we can start to compare some of the structural properties of the network with those different iterations. So just to talk through some of this, we have 4 different sort of structural properties of the network, and you can see these black lines that essentially represent these simulations where we've randomly swapped the nutrients between different prey. And then you can see the little blobs that essentially tell us what the structural property is of the actual network that we looked at. And I won't go into too much detail with these network metrics, as I realize they're a little bit impenetrable. But just to talk about a couple of them, linkage density on the right there is essentially just how much is going through the network. So how many interactions are going on per the number of species in the network. And what we see is, based on our random simulations of swapping things, we would expect carbohydrate to be a relatively high linkage density, but actually it's quite low, which means there's not much carbohydrate going through the network, but also a lot less than we'd expect if the spiders were randomly foraging. And carbohydrate, a bit like with us, we don't want to eat too much sugar. Spiders as well can have all sorts of fitness impacts if they spend too long eating sugars. And we can see that the protein in the top right there, so the red blob, is actually bordering on slightly higher than we'd expect. So they're clearly foraging quite intensively for that protein. And I thought modularity would be an interesting one to talk about as well, which is basically how separated the network is. So can we just sort of separate it into different blobs? If we were to cut the network with a knife, can we just separate it without cutting any of the links? And what we see is that carbohydrate is very low, much lower than we'd expect, which means that actually the carbohydrate is really well joined up across the network. There's not much separation going on. But how do pests factor into all of this? And we can start to look at the network properties based on pests versus beneficial organisms compared to the whole network. So if we just look at the interactions between spiders and pests, we start to see some really interesting things. So generality, which is just how much nutrient the spiders are getting for each of them, we can see that that's quite low for beneficials and relatively high for pests. They're getting quite a bit of lipid and protein from their pests, which is interesting. And then vulnerability, which is the opposite, so how much protein is leaving the prey essentially, we can see is quite low for beneficials again, but actually higher for pests than it is for the network generally. So a lot of the nutrients are essentially coming from those pests. And I think one of the most interesting bits is modularity. So we can see that, I'm just going to move that toolbar. So we can see that for beneficial invertebrates, so that could be pollinators, predators, anything that we'd consider to be doing us a service, is quite high modularity. So that means the network's quite well separated. Whereas for pests, it's very low. So that means that there's a lot of crossover in the interactions for particularly protein for these pests. So that's possibly indicative of some interesting competitive effects. So the question that naturally comes from that are spiders competing for specific nutrients? And just to show you another representation of this network, we have all of these individual spiders. So in this case, we're not looking at spiders as species, we're looking at them as individuals. So each of the light grey circles is an individual spider, and each of the dark grey circles is a different prey group. And we can see that there are essentially just a lot going on. There are all sorts of different interactions, but we can start to look at competition by looking at interactions between spiders for the same prey. So if two spiders are eating the same prey in the same place at the same time, we can infer from that they're competing. So we can actually draw a link between them that we would call a hyperedge. And we can even just remove the links with the aphid sort of individually and just focus in on that one interaction that tells us essentially how much these spiders are likely to be competing. And we call this type of competition exploitation competition, where the spiders are sharing the same prey. And if we draw that onto our network, it starts to look even messier. So again, we can remove those interactions with the prey and just focus in on those bits of competition where the spiders are sharing the same prey in the same place at the same time. And we can essentially see from this that there's a lot going on. But to focus in on some really hot off the press results that literally came out in the last couple of days, we can actually see here in this ternary plot again, where we're seeing the points based on the nutrient content. The spiders that are eating quite lipid-rich diets and quite carbohydrate-rich diets tend to have a higher frequency of competition. So they're competing more when they're eating lipid and carbohydrate-rich food, which is super interesting. And we can see on the right what this network might look like as a nutritional network of competition. So we're really getting into the sort of complex nitty-gritty now. But essentially, from this analysis, we're starting to see that all three nutrients are driving competition, and particularly the sort of ratio of protein to lipid is really important in determining which prey spiders are competing for. So one of the next questions that we want to start answering with this nutritional network concept is focused on network robustness. So essentially, especially in the Anthropocene, where all of our ecological systems are being hammered by different pressures, whether that's artificial light, pollution, climate change, you name it, we can start to look at whether our ecological networks are going to be resilient to those perturbations. So within a nutritional network context, we're interested in whether the flow of nutrients is sustainable with these different pressures, and how the flow of nutrients is likely to change when we do put these pressures on the network, and how that's going to change the structure of the network specifically. So we would refer to the resilience of the network, as I said, as the robustness. But just to give you an example of that, so this is perhaps my sleep deprived brain, sort of drawing an analogy from reading stories to my 9-month-old son. But I wanted to focus on an example from Dr. Seuss's The Lorax, which hopefully many of you will know about. Anyone who hasn't, it's an ecological dream as a text. But within the story, there are basically these wonderful trees called truffula trees. So you can see these sort of candy floss looking trees at the center of this network. And we have swammy swans that sort of nest in and around the trees. We have barbaloot bears that feed on the fruits, and we have the humming fish that bathe in the rivers that are sort of under the trees. And then this Onceler character comes along and wants to deforest the trees to make some weird clothing. But in this case, we've got solid links between Onceler and the barbaloots with the truffula trees, because they're directly exploiting them, and these little dashed links between the swammy swans and the humming fish and the truffula trees, because they're just using it for habitat. But if as happens in the story, sorry for the spoiler for anyone who's not familiar, the Onceler does deforest the truffula trees, essentially all of those nodes are losing their only connection. So they in turn are also lost. They have nothing left to interact with. So we refer to that as a secondary extinction. So in terms of ecological network robustness, this network is not very robust at all. Just one perturbation event completely topples it. But if, for example, the barbaloot bears were managing to forage on green eggs and ham as well, so diversifying the things that they're eating, and perhaps them and the swammy swans can not just depend on the truffula trees for habitat, but also maybe go into Whoville from the Grinch. And perhaps the humming fish can go and live with the one fish, two fish, red fish, blue fish in whatever place they're living in. Then as soon as we lose the truffula trees, actually all of these nodes, except for the Onceler, can survive. So in that case, we have a more robust network. So essentially, the thing I wanted to illustrate with this is having more interactions is essentially what makes a network robust. But in terms of a nutritional network, we start to get a completely different context. So if we lose the bottom right resource node, so this one that you can see that's now sort of shaded out, you can see the top right consumer, which might again be a spider, is completely lost because it has nothing to interact with. So that's a bit like the example of the Lorax network that we just saw. But you can also see that sort of second from the top right spider, its nutritional proportions have changed, so it's now eating more carbohydrate and less lipid than it was before, because it's lost this really nice lipid-rich resource. So we can start to see how losing nodes might actually change the structure of this network and the nutrients that the spiders are getting. And if we lose this bottom left one, although this top left spider still has two things to interact with, you can see it's lost almost all of its carbohydrate. So that might mean that spider can't survive. So we can start to understand how these networks of interactions can survive perturbations from the perspective of nutrition. So we might see that node completely lost in this case, or we might see that it starts to instead eat something else that's carbohydrate rich to replace the thing it's lost. So using this nutritional network approach, we can start to understand and predict how these networks are going to change over space and time, especially as we devastate our ecosystems. And there are some really exciting examples coming together. So I've been working with B. Dale, a PhD student over in Northumbria, on these sort of nutritional networks of pollen and nectar for pollinators. And we've seen that pollen provides a lot of protein to the foragers, whereas nectar provides not just carbohydrate but also a lot of lipid in a lot of cases. And how these two nutritional sources help the pollinator community is really changes over all these different sort of species, and particularly looking at the role of trees in these networks has been really exciting. But we're also, a bit like that example from Timberlake et al. that I mentioned earlier, we're starting to explore how these nutritional networks might start to influence the lives of people as well. So we just had this paper published in Trends in Ecology and Evolution, which shows how sort of invasive fish might completely change the nutrients available to people in sort of fishing communities across the world. And we can start to apply these ideas to entomological contexts as well, particularly by thinking about that example from Timberlake et al. where the nutrients are provided as a result of those pollination interactions. So there are a few steps remaining for nutritional networks. We want to develop new ways of modeling these interactions, particularly thinking about how those interactions change in response to perturbation. We want to look at how these networks rewire, so how those interactions might switch based on nutrients. So are these animals going to interact with different things but still manage to balance their nutrient intake? And we also want to really develop this idea of looking at the robustness of nutritional networks, which is really exciting and hopefully something we can start to make a lot of progress on very soon. So just to highlight, we're doing some really exciting nutritional work. So we in my group we have 2 PhD students doing some nutritional research at the moment. So Mia Croft is looking at how artificial light alters invertebrate nutritional networks, and Rosie Christopher's looking at how nutrients drive commensal interactions between invertebrate predators and plants. So hopefully through those examples as well, we'll start to really understand how these nutrients are driving a lot of these really exciting ecological dynamics of invertebrates in the field. But thanks so much for listening. It's been really great to talk through this and hopefully interesting for all of you. And if anyone has any questions, I'd be really excited to answer them, and always happy to chat more about this and everything else sort of related to this. Thanks so much.